Neurons communicate through electrical signals that enable rapid transmission of information across the nervous system. Understanding the electrical characteristics of neurons is fundamental to comprehending how the nervous system functions at a cellular level. At rest, a neuron maintains a resting membrane potential, typically around -70 mV. This electrical charge difference across the neuronal membrane arises from the unequal distribution of ions, mainly sodium, potassium, chloride, and negatively charged proteins. The membrane is selectively permeable, allowing potassium ions to move more freely than sodium ions, and the sodium-potassium pump actively transports 3 sodium ions out of the cell and 2 potassium ions into the cell, maintaining the ionic gradients. When a neuron is stimulated, its membrane potential changes. If the stimulus is strong enough to depolarize the membrane to a threshold level, usually around -55 mV, an action potential is triggered. This is an all-or-none electrical impulse that travels along the axon without decreasing in strength. The action potential involves a rapid sequence of ion channel openings and closings. Voltage-gated sodium channels open first, allowing sodium ions to rush into the cell, causing a rapid depolarization. Shortly after, sodium channels close and voltage-gated potassium channels open, allowing potassium ions to exit the cell, repolarizing and even briefly hyperpolarizing the membrane. This restores the resting membrane potential. The action potential propagates along the axon through a process called saltatory conduction in myelinated fibers, where the impulse jumps between nodes of Ranvier, speeding up transmission. In unmyelinated fibers, the action potential travels continuously along the membrane. When multiple axons in a nerve are activated simultaneously, their individual action potentials summate to produce a compound action potential. This is the summed electrical activity recorded from a bundle of nerve fibers. The compound action potential reflects the number of fibers activated, their conduction velocity, and synchrony. It is a valuable measure in electrophysiological studies and clinical nerve conduction tests. Neurons communicate with each other or with effector cells at specialized junctions called synapses. Most synapses are chemical, involving the release of neurotransmitters. When an action potential reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. Calcium influx into the terminal causes synaptic vesicles filled with neurotransmitters to fuse with the presynaptic membrane and release their contents into the synaptic cleft. The neurotransmitters then diffuse across the cleft and bind to specific receptors on the postsynaptic membrane. Depending on the receptor type, this binding can cause excitation or inhibition of the postsynaptic neuron by opening or closing ion channels, leading to depolarization or hyperpolarization. Electrical synapses, though less common, allow direct passage of ions through gap junctions, enabling faster, bidirectional transmission. Synaptic transmission is modulated by several factors including neurotransmitter reuptake, enzymatic degradation, receptor sensitivity, and synaptic plasticity. These mechanisms play crucial roles in learning, memory, and adaptive responses. In summary, the electrical characteristics of neurons involve resting membrane potentials, generation and propagation of action potentials, and the summation of signals as compound action potentials. Communication between neurons occurs mainly through chemical synapses, where neurotransmitters mediate signal transmission. This intricate electrical and chemical interplay forms the basis of nervous system function, enabling everything from simple reflexes to complex cognitive processes.